Yeah, hi, everybody. I just have there are about 11 or 12 digestive questions, surely less than 15, on the final exam. I realize it's not until Friday, but I might as well share some information that you might appreciate with regard to that. So let me give it to you now. Okay. And that is basically, they're physiological questions, obviously, but the whole idea is where do the different enzymes are active and what parts do what things. So in the mouth and the esophagus, there's really no absorption. The absorption effectively really doesn't start and effectively almost concludes in the small intestine. There's a limited amount in the large intestine. In the stomach, maybe some, but generally it's problem things like alcohol and caffeine and certain chemical agents that are typically GI irritants like nonsteroidal anti-inflammatories and things like that. So in the mouth, we have salivary amylase, lingual lipase. That's it. Esophagus, none. Stomach, we begin to start proteins. That's where pepsin or the pepsinogen activated by pepsin in the presence of hydrochloric acid and augmented by gastrin and histamine and acetylcholine from a variety of sources. And there may be just a tiny amount of gastric lipase as well. So effectively, we may have started the digestive process on carbohydrates, fats, and proteins, but they haven't really completed by any stretch of the imagination. And we have neither started nor certainly finished the nucleotides. And those are the four real groups of any impact. So that's really the jumping off point. It's all about the small intestine. The small intestines are interesting because we start being a very acidic environment from the chyme shooting in, and by the time we end, we're in a very alkaline environment. And it's those pH changes, and it's the osmolality and the hyperosmotic material, and the high concentration and the high degree of acidity, along with stretch receptors, that triggers uh, a variety of changes when it gets to the small intestine, particularly the release of secretin and cholecystokinin. Cholecystokinin plays a pivotal role in fats, releasing bile, and also playing a role with regard to pancreatic aspects. And the secretin really releases all that slew of pancreatic enzymes. And that's really the big player. While in the stomach, when food enters, and it's mostly stretch receptors there, that triggers the release of the pepsinogen and the hydrochloric acid, the intrinsic factor, eventually pepsin forming, and then those enteroendocrine cells, particularly for gastrin, play such a pivotal role in working and amplifying what the stomach is doing on the inside. So those are some key features. So really, most of the nearly for our purposes, really all the absorption, with the exception of maybe some B vitamins and some K vitamins and the large intestine, and maybe some water and some electrolytes to kind of balance things out. Pretty much all the absorption occurs in the small intestine. And so, for instance, and really the big players bringing it down to the molecular level that are the smallest possible unit, where are called brush border enzymes. They are really the big players that really allow things to be absorbed. Okay. So, for instance, if we have sucrose goes to glucose, fructose, and lactose, glucose and galactose, and maltose just breaks up glucose long chains. Particularly find them in things like, in long chain carbohydrates like glycogen, like starches. But once they break them down to the component parts, be it glucose, fructose, galactose, whatever they are, they're absorbed by way of the brush border enzymes, really are the ones that break it down to that final monosaccharide and allow it to be absorbed. So they work that way. And that structure that's located in the intestinal villi, and particularly the villi and surrounded by the microvilli, are that combination of a lacteal for fats and heavier products, and then surrounded by a capillary bed that absorbs the water soluble things, a la carbs that are there. So I think that those are really important factors to remember. You should know where the enzymes are produced. So whether it's varying kinds of amylase or various protein digesters or fat digesters, carbohydrate digesters, know where they're produced and know where they're active. So that's something else you need to know. So really the big player in this is the small bowel more than anything else. All the activities that go on there, the movements, which are, and all that immense amount of surface area from the plica circularis, the circular folds, the villi, the microvilli, the brush border enzymes, all the other pancreatic juices and the bile salts that work so well with lipids, and the pH that gradually changes from acid to alkaline are all really big players. So the villi themselves, which are just millions, literally, they all have that lacteal in the capillary bed and in the brush border, and so all of those are part of it that are there. So I think that's an important factor as well. And let's see. Uh, the key issue is certainly the entry point, which we call the common bile duct. So that has a combination of bile, which is about fat, bile salts, and certainly some waste products, along with what's coming from the pancreas, which are most of the enzymes. And they come that way first by the pancreatic duct and the hepatic or cystic duct, and eventually become the bile duct. And when they join together, it's the common bile duct or hepatopancreatic ampulla, they want to call it. And other factors that are important, the large intestine, even though it's not essential, does play a really important role in uh, really finalizing things, and the vitamins like B and K, electrolytes and water. And the advantage that it has is that it can be reseeded in case of colitis or some other significant infection that causes like profound diarrhea from any cause that's there. And we're fortunate because really the current feeling is the appendix is sort of a warehouse for this that will repopulate it. And of course, using things like yogurt and probiotic agents and things along that line that are inherently have a lot of that material in them. So I think that's really the critical feature. And again, we always call them collectively all those enzymes zymogens because we activate them, whether it's by acidity or alkalinity or a cascading reaction. So they're really important that are there. And let's see if there's anything else. And by and large, what we see with these, uh, particularly when you look closely into how things fats get absorbed fairly easily because they cross the membrane. Most of the other things involve a combination of facilitated diffusion, and very often they use the anti or counter transport mechanisms associated with sodium. Sodium kind of moves in and out actively, and then we begin to use a variety of different things. So I would check closely to see exactly how things like disaccharides, monosaccharides, fats, etc., that are absorbed in the small intestine. So take a look at that very closely. I think that covers just about everything that you need to go over. See, I think everything else is urinary or gastro or male and female repro, and some of the developmental stuff should also be on there as well. As I'm just scrolling through to make sure everything is there. Yeah. Again, the additional questions on digestive are certainly to understand the roles of which ones are proteolytic, and I want to amplify that again, which ones are fat breakers, like amylase for starches and lipases for fats, and then a variety of peptidases of many different types, and the roles that they play. So thanks very much. I think that's everything. If you have any questions, feel free to email me, and I will be in on Monday for Science Day, and you may be able to catch me I'll be in for a couple of hours. So thanks very much, and bye-bye.